CRISPR-Cas9 is one of the most revolutionary scientific discoveries in recent years, earning its discoverers a Nobel Prize in 2020. With this technique, scientists can easily and accurately edit DNA sequences, opening up all sorts of possibilities for treating genetic diseases or improving crops. But it's also sometimes called genetic scissors or molecular scissors. So how does it actually work? Well, we made a video to explain. Let's cut to the chase. DNA is a long polymer molecule. It's like a chain that contains instructions for building and maintaining an organism. These instructions are encoded in the sequence of 4 chemical bases: adenine, guanine, cytosine, and thymine, which are represented by the letters A, G, C, and T. The sequence of these bases determines what traits an organism will have. For example, the gene for eye color has different variants that result in different shades of brown, blue, green, or gray eyes. If we know which genes are responsible for certain traits, then in theory we should be able to modify them to prevent diseases or change those traits in other beneficial ways. But DNA isn't just a single strand. Instead, it's twisted into a double helix with 2 strands that run anti-parallel, meaning they run in opposite directions. Each base on one strand pairs with a complementary base on the other strand. Adenine always pairs with thymine, and guanine always pairs with cytosine. Because these base pairs are so specific, we can use this knowledge to break apart the 2 strands of DNA with complementary base sequences. And if we know the sequence of one strand, we can predict the sequence of the other strand simply by substituting A for T and vice versa. This complementary base pairing is important for understanding how CRISPR works because it allows us to target specific sequences of DNA for editing. The word CRISPR might sound like a new type of haircut, but it stands for clustered, regularly interspaced, short palindromic repeats. That's not exactly a household word either. So let's start with the part that's not CRISPR. Cas9 is an enzyme that acts like a pair of scissors. It cuts DNA at a specific location. CRISPR are sections of DNA that have a specific sequence of bases that are repeated over and over again. Some scientists think that these repeating sequences were originally a defense mechanism against viral infection. As viruses attack and inject their own DNA into cells, some of that viral DNA ends up getting incorporated into the CRISPR regions of the cell's DNA. Over time, these incorporated sequences build up, and the cell can recognize them as foreign, triggering an immune response. Scientists believe that this is how CRISPR systems evolved, and today they're still used by bacteria as a sort of immune system. But when scientists realized that they could use these bacterial defense systems to cut DNA at specific locations, they started calling the whole package CRISPR-Cas9. Here's how it works. First, we need to direct Cas9 to the right location in the genome. To do that, we give it a guide RNA, a short piece of RNA that's complementary to the DNA sequence that we want to target. Cas9 then uses this guide RNA to find the matching DNA sequence in the genome. When it finds the right spot, Cas9 makes a cut. It's important to note that we're not trying to change the sequence of the DNA here. We're just making a cut in the DNA strand. You may be wondering why we'd want to do that. Well, DNA has the ability to repair itself. It's actually constantly repairing itself from damage that occurs naturally over time. Cells have mechanisms for detecting damaged or incorrectly paired bases and excising them, then replacing them with the correct bases. We can take advantage of this natural process by providing our own template strand of DNA with the correct sequence. If we provide a template with a different sequence, the cell will use that to repair the DNA. In other words, we can use Cas9 to cut the DNA and then provide a new sequence that the cell will incorporate into the genome. This process is what we call gene editing, and there are many ways we can make use of it. Sometimes we want to knock out a gene, which means preventing it from producing its protein product. This can be useful for studying the effects of a gene or for eliminating a gene that's causing a disease. In this case, we would choose a location within the gene to cut. Then, when the cell repairs the DNA, we provide a template that introduces a premature stop codon into the gene. Stop codons are sequences of 3 bases that signal the end of a gene. By introducing a stop codon early in the gene sequence, we prevent the cell from producing a functional protein. Other times, we may want to introduce a new gene into the genome. In this case, we would choose a location near the gene we want to insert. Using the same method, we would then provide a template that includes both the gene we want to insert and a short sequence that's complementary to the DNA on either side of the cut. Since the cell doesn't know where the new gene came from, it will incorporate it into the genome and treat it as if it had always been there. Sometimes we may want to make a small change in a gene sequence without knocking out the whole gene. In this case, we would choose a location within the gene to cut, then provide a template with the desired sequence. This way, the cell will repair the DNA and incorporate the changes we want to make. For example, one of the most common genetic disorders is sickle cell anemia, which causes red blood cells to become misshapen, leading to all sorts of health problems. Sickle cell anemia is caused by a mutation in a gene called HBB, which codes for a protein called hemoglobin. Hemoglobin is responsible for transporting oxygen in red blood cells. The mutation causes a single amino acid change in the protein, which causes the red blood cells to become rigid and sickle-shaped. This makes it harder for the cells to move through small blood vessels and can cause blockages in the blood flow. It can also lead to pain, fatigue, and organ damage. Researchers are working on developing a cure for sickle cell anemia using CRISPR-Cas9. One approach is to use CRISPR-Cas9 to knock out the mutated copy of the HBB gene on one chromosome and then insert a corrected version of the gene. This would restore the production of normal hemoglobin and prevent the sickling of red blood cells. Another approach is to use CRISPR-Cas9 to make a small change in the DNA sequence of the HBB gene. This would correct the mutation without knocking out the whole gene. This approach is known as base editing. Base editing has the advantage of being more precise than traditional CRISPR-Cas9 because it doesn't require a template strand. This means that there's less risk of introducing unintended changes into the genome. While there's still more work to be done, researchers are hopeful that CRISPR-Cas9 base therapies will soon be available for people with sickle cell anemia and other genetic disorders. In addition to curing genetic disorders, CRISPR-Cas9 can also be used to develop new cancer treatments. One approach is to use CRISPR-Cas9 to knock out genes that are involved in the growth and spread of cancer cells. This can help to slow or stop the growth of tumors and make them more susceptible to chemotherapy. CRISPR-Cas9 can also be used to engineer immune cells to better fight cancer. For example, researchers are developing a type of immunotherapy that uses CRISPR-Cas9 to modify T cells. T cells are a type of white blood cell that plays an important role in the immune system. They can identify and destroy cancer cells, but they can also become exhausted and stop working effectively. Researchers are using CRISPR-Cas9 to modify T cells to make them more resistant to exhaustion and better at fighting cancer. CRISPR-Cas9 has also been used to develop new diagnostic tools for infectious diseases. For example, researchers have developed a test that uses CRISPR-Cas9 to detect the presence of SARS-CoV-2, the virus that causes COVID-19. The test is much faster and more accurate than traditional methods, and it can be used to diagnose patients in resource-limited settings. One of the most exciting potential applications of CRISPR-Cas9 is the development of new gene drives. A gene drive is a genetic element that can spread rapidly through a population. Researchers are developing gene drives that can suppress the populations of disease-carrying mosquitoes. This could help to control the spread of diseases like malaria and Zika virus. As you can see, CRISPR-Cas9 is a powerful tool with a wide range of potential applications. While there's still more work to be done, researchers are hopeful that it will revolutionize the way we treat genetic disorders, fight cancer, and combat infectious diseases. Now watch how genome editing could help save endangered species, or check out another one of our videos.